own beautiful moments, and this is one of those. In this tough time, we are more free to design and conceive different ideas, and to implement the same we have digital platform, and that too in a very convenient way. In a physical form, if you want to have a workshop of this kind, you need at least rupees 5 lakhs to organize this kind of workshop. Here, without compromising the quality of the workshop, you can have this much of number, and this is really wonderful. And for this, I personally congratulate Professor Sunat Tiwari to conceiving and for delivering this kind of workshop.
Thank you
As far as
so
the
much.
topics are concerned, topics are really beautiful. Small topics, but very relevant, very important. That's the key thing. Usually, people don't think and don't conceive such type of topics while delivering online on e-learning platforms. But the kind of topics which I am seeing personally, this may attract a person from another field also, and I am hoping that person from other field also might have joined this particular workshop of biotechnology, because this is a time where we should learn about the DNA, we should learn about the genetics engineering, we should learn about how these things works and how can we defeat this virus of COVID. This is the key thing, and I am hoping that RGPV will be doing something wonderful, which in turn will be remembered by in the history of the university, wherein we have contributed in some way or another way in defeating this COVID virus. Thank you very much for inviting me as a chief guest and to say a few words in this particular workshop. I hope this workshop goes on for 5 days and brings results and laurels to the university. Thank you very much.
A very big thanks from the bottom of our hearts to our Honorable Vice Chancellor, sir, for your stimulating words. Now the time to begin the workshop. A pleasant evening to all of you. Small cheer and great welcome makes a merry feast. Cordial welcome to all the participants of this workshop. Shubham Karotu Kalyanam, Arogyam Dhan Sampada, Shatru Buddhi Vinashaya, Deepajyoti Namastute. Salutations to the light of the lamp, which brings auspiciousness, health, and prosperity, which destroys inimical feelings. We are very fortunate to meet today in this momentous event in which overwhelming response of the participants from all over the world, which is beyond imagination. More than 600 participants have registered for this workshop. The eligibility of this consortium workshop was graduation and post-graduation. But we are very happy to inform you that the doctoral fellows and the faculties of the various institutions, head of the departments, have also registered for this workshop. We are also heartily welcoming all of you. All the participants of this workshop have been divided in 3 batches. We hope all have received their links for joining this workshop. The main objectives of this workshop are: number 1, focus on the fundamentals of the lab experiments, where usually the people are doing silly mistakes. The second objective of today's workshop is some institutions are running the biotechnology course, but either they are not having the lab facilities or not having the trained or specialized faculty. So, through this platform, we are trying to give some glimpses of the biotech culture and real or actual flavor of the subject. Hence, this Consortium Biotech Workshop is a series of 5 workshops on key aspects of biotechnology, other biological sciences, and somewhat computer sciences along with IT. Keeping all these facts in our mind, School of Biotechnology, an autonomous university teaching department, Rajiv Gandhi Pradyogiki Vishwavidyalaya, University of Technology of Government of Madhya Pradesh, accredited with grade A by NEC, Bhopal is also going to organize hands-on training on the same series of workshops which we are organizing on the digital mode. Those who are really interested, register themselves on the link given below on their e-certificates generated after the completion of each workshop. Now, we are focusing on the today's workshop, that is how to start the lab work under aseptic condition. Whenever we are talking about the aseptic condition, today's workshop has divided into 4 sessions. Session 1, based on fumigation. Session 2, cleaning of the glasswares and sterilization of their glasswares and chemicals. Session 3, preparation of the solutions, stock solution and working solutions, weight by volume or volume by volume. And finally, we have the fourth session, that is the feedback session. These 4 sessions we have divided in 2 days, June 1st and June 2nd. 2 sessions on each day. First, second sessions we are doing today on 1st of June. Third and fourth session we will do tomorrow on the same platform at the same time. Now, come to the first session, that is first session of the first workshop, that is how to start the lab work under aseptic conditions. Everybody is knowing we are already locked down or quarantined in our homes from the last more than 2 months. All offices, all labs, all institutions are locked down. N number of work has already been piled up. Now, again we have to restart our work. Again we have to restart our labs, departments, etc. Then what we have to do? Whenever we are reaching to our labs, is it possible to start our work immediately? Not. Why? Because after a long time, that labs are not under use. So n number of contaminants are present there. How can you say the contaminants are present there? For this purpose, our first session of today's workshop is session 1, that is fumigation. This fumigation is comprising of 3 steps: pre-fumigation, fumigation, post-fumigation, then only the aseptic condition occurs. Because we are not in a position, why we are not in a position to start our lab work? Because our labs are not having aseptic condition or aseptic environment to start our lab work. If we are starting our lab work under such condition, our all experiments will be failed. So, always keep this fact in your mind. Whenever you are starting your lab work, your lab should be started under aseptic conditions. Again, question arises in our mind, why the need of pre-fumigation? The reason behind this is requirement of microbiological cleanliness. If any of the microbes, which are n number of microbes which are present in our lab, they will create hindrances or they will create contamination in our experiments. So, with the help of gaseous sterilization for killing of the microorganisms, then only after checking of all these, then only we are in a position to start our work. Along with this, prevention of microbial growth in air, surface of wall or floor, particularly of the lab. These are the important things. That's why there is a need of pre-fumigation. Excuse me. Then what is the significance of pre-fumigation? Just to check the significant reduction in most of the isolates. Never be the labs are 100% free from microorganisms or isolates. 99.99% the germs or the microbes are removing. But no one lab in this world is 100% free from contaminants. To the predominant microbial isolates, these predominant microbial isolates are, which are usually present in our lab, these are Klebsiella pneumoniae, Escherichia coli, Staphylococcus aureus, Pseudomonas aeruginosa, Streptococcus faecalis, Acetobacter species, Coagulase negative Staphylococci, and Syringia species. These are commonly present in the labs as the contaminants. For instance, we have selected the School of Biotechnology, an autonomous university teaching department, Rajiv Gandhi Pradyogiki Vishwavidyalaya, University of Technology of Government of Madhya Pradesh, accredited with grade A by NEC. The glimpses of some labs which we have to check whether these labs are free from contamination or not. For instance, the lab of genomics and genetic engineering, environmental and bioprocess engineering lab, animal tissue culture lab, plant tissue culture lab, microbial culture lab. Because the students are working, it means they are working under aseptic condition. But after too much or this much long gap, our labs are totally dumped. Now we have to just again restart that work and again restart our working culture there also. For that purpose, there is a need of pre-fumigation. What is pre-fumigation? First, we have to check whether these labs are really need of fumigation or free from contamination, or these labs are already ready. As we know very well that nowadays all labs are totally contaminated. First, we have to check all these labs where the contamination is present, which type of contamination is present, what is the load of contamination which is present in various labs. This is the laminar flow, everybody knows. In this laminar flow, we have to prepare our media, autoclave it, and keep inside the laminar flow just to cool down up to 50 to 55 degrees centigrade. Why 50 to 55 degrees centigrade? Because agar solidifies at 40 to 45 degrees centigrade. We are taking the nutrient agar to check the microbial growth of the various organisms. Why we have selected the nutrient agar? Because it mainly covers the wide range of microbes for their growth. Media is prepared, and this media is usually prepared in the conical flask. Whenever we are preparing the conical flask, preparing the media inside the conical flask, always hold the flask from the bottom. Why hold from the bottom? It provides the grip. You can easily do your experiments with the help of when you are just holding the flask from the bottom instead of neck. What will happen? Sometimes you are holding the flask from the neck and your media is warm. Immediately it is falling down and your beaker is breaking, as well as the media which is present inside that beaker is spilling out and totally spoiled. Just to avoid such type of stage, we have to just hold from the bottom. Now our media is prepared. This media is nutrient agar, right? Suppose we are preparing the media which do not contains agar, then what will happen? That is known as broth. And such media which contains agar, that is known, and that is pouring in the petri plate, petri dish, and converting that petri dish into the petri plate. This is the petri dish which do not contain any of the media. That is blank vessel or blank petri plate made up of glass or fiber. This is known as petri dish. And once we are pouring the media inside that dish that is converted into the Petri plate. How we are preparing this nutrient agar? 2% of nutrient agar in 20 ml of distilled water, which is suitable for the preparation of one plate. Calculate this as per your requirement. Suppose you are going to prepare 20 plates, so multiplied by 20. Sure. Then our plates are ready. We have prepared all these plates inside the laminar flow. Nowadays, do not use the spirit lamp inside the laminar flow. Why? Because the principle of this laminar flow is based on 2C principle, that is counter current. What is counter current? This is the laminar flow blower and we are sitting here. This is the blower. We are sitting here for our work. Then what will happen? When we are using the blower at the time of our work, that air comes out from that blower to us, opposite to each other. That's why it is known as counter current. There is already this aseptic condition is available here. Aseptic atmosphere is available here. That's why there is no need of a spirit lamp to create or to develop more and more aseptic condition. No need. Our plates are ready, right? We have prepared the media, that is nutrient agar media. 2% of nutrient agar dissolved in 20 ml of distilled water. Autoclaved it. Keep at the room temperature inside the laminar hood to cool down up to 50 to 55 degrees centigrade. As we know, agar agar solidifies at 40 to 45 degrees centigrade. In this manner, we have prepared our plates. Now, how to keep the plates? Always on the bottom up position. Why always on the bottom up position? Two reasons behind this. First one is to prevent condensation, which often accumulates on the lid of the dish during incubation from dripping down on the culture. Whenever we are pouring at the time of the because our media is slightly warm, vapors are coming out and they are depositing on the lid by the process of condensation. Just to prevent condensation whenever we are pouring our media into the Petri dish and that dish is converted into Petri plate. And second important reason is to provide maximum surface for the growth of the microbes aerobically and anaerobically. These are the two important reasons why we are always keeping the Petri plates on the bottom up position. Whenever we are doing any of the microbiological work, always keep the plates on the bottom up or inverted position. Never be on the top up position. Now our plates are ready. We have to check the various labs of the School of Biotechnology, whether they are really ready to start our work under aseptic condition or not. For this purpose, we have to keep the way on various places of these plates, above the equipment, inside the equipment, on the floor, above the bench, working bench, inside the drawers, inside the table, inside the on the shelf, everywhere. Not only in one lab, but in all labs, right? Because where the contaminant is present, we cannot say. So we have to just keep all the plates in the bottom up position to various places of the various labs of the School of Biotechnology, for instance. Once we have selected the medium, once we have selected the place where we have to keep all these plates, now 4 important characteristics of the pre-fumigation. First is seal that area where you are doing your pre-fumigation. Demarcate that area. This is the area where we are doing the pre-fumigation. Number two, just inform the concerning staff or the concerning officer of that school that this lab is contaminated. We are going to check this lab for pre-fumigation. Third, just paste the notice on that lab. This lab is contaminated. Do not start your work because this lab is not having aseptic environment. And number four, only trained person will do such type of work. All ventilations should be off of that lab, just to provide the spreading of the contamination to other labs. Right? What are the four important characteristics of pre-fumigation? Number one is sealing of the area where we are going to do the pre-fumigation. Second, ventilation is totally off. Third, notifying, just pasting the notice on the lab. And fourth, informing the staff or the officer of that institute or the lab or that department if this lab is not ready to start our work under aseptic condition. We are keeping these plates for incubation of 24 to 36 hours. Why 24 to 36 hours? Because some microbes are showing their growth up to 24 hours, but some microbes are growing very, very slow. That is, their growth rate is very slow. That's why 36 hours are required. Now, our incubation period is over. The time is for the observation. Now, where we have kept all these plates, we have to just check. We kept in this type of plates that is totally plain, totally clean. There is none microorganism or nothing is present except the nutrient agar. But when we are observing, oh, we are getting the contamination, we are getting the colonies. Oh shit, our lab is not ready for the any of the lab work. How to start our lab work? Either the contamination is of this type, or any of these types. What we have to do? If we are getting any of these types contamination, it means we are not getting such type of contamination to go ahead to start our lab work. What we have to do? If the contaminations are present of any of these types, now there is a need of fumigation. Here, I am requesting Riddhi Tiwari, Doctoral Fellow of School of Biotechnology, Rajiv Gandhi Pradyogiki Vishwavidyalaya, to throw light on the process of fumigation. Over to Riddhi Tiwari.
Thank you so much, ma'am, and a warm welcome to all the participants of this workshop. I thank you all from the bottom of my heart for showing your enthusiasm to join and show your interest in this consortium biotech workshop. So as ma'am rightly said, that once we have checked that our labs are not fit for our work, so the need for fumigation arises now, because we have been in the lockdown because of this COVID-19 pandemic for a long time now, and our labs have been closed, and surely there might have been a lot of contamination in those labs. So it is our utmost responsibility that before using our labs for any sort of work, it is required that we fumigate our labs to make sure that there is no trace of any contamination or infestation whatsoever in those labs. So coming to the most commonly used material for fumigation is formaldehyde. The reason behind using formaldehyde is that it is a broad spectrum disinfectant. That means it has the ability to kill most of the bacteria, viruses, fungi, and the most notorious microorganisms like endospores. These endospores might be the cause of a lot of contamination. So the formaldehyde helps in removing all of these microorganisms, that too at low temperatures, like for example, when we sterilize, like other speakers will be telling you in the coming session about sterilizing using hot air oven and autoclave. In that we need a lot of high temperature, but fumigation can be carried out at low temperatures. Why do we use formaldehyde for fumigation? Because this is the most well-known method which is currently being used in India and many parts of other and many other parts of the world. It is used wherever microbiological cleanliness is required, be it not only our labs, it is regularly used in operation theaters, in the hospitals, as well as in poultry farms, even in all sorts of veterinary establishments also. So why do we use formaldehyde? The second most important reason is it is the most cost effective procedure that is available. I'll be coming to other alternatives also, but this is the most commonly used method, and it is the most cost effective method to bring about 99.99% cleanliness, microbiological cleanliness in the labs, which we are going to use after this lockdown. Uh, if we talk about the physical properties of this formaldehyde, it is a colorless gas at room temperature, having a peculiar, characteristic, pungent smell. As we have seen in the first slide, that fumes are generated in fumigation. So hence, this is a gaseous sterilization. The three important steps that need to be followed while performing fumigation are three steps. First of all, we need to clearly seal the lab area properly so that when fumigation process is being carried out, none of the gases percolate outside of the environment, which is, or the area which is being fumigated. Next step is the fumigant is then released into the lab area thoroughly to each and every corner of the area or the room where we need to fumigate, so that it percolates throughout the space and removes any sort of infestation or contamination whatsoever. Finally, last but not the least, ventilation of that area is of the most important aspect, so that before anybody enters into the lab for working, all the poisonous gases are completely removed from that space so that it is safe for the humans to use. What is the method of fumigation? Most commonly used method of fumigation is that we mix 150 grams of potassium permanganate with 280 ml of formalin, that is 40% aqueous solution of formaldehyde. This formula is made when we are going to fumigate around 1000 cubic feet area of the lab. For this, the fumigant is placed in different bowls at different places in the lab so that each and every nook and corner of that lab or that area is fumigated properly. Next comes the important aspects while carrying out the fumigation. The person who is going to carry out fumigation must be using proper personal protective equipment, that is PPE, that includes caps, masks, gloves, foot covers, spectacles, and any other protective gear so as to protect himself or herself from the poisonous gases which are being prepared while fumigation. This is because we all know formaldehyde is not only an irritating element for your eyes or nose, but also it is a potential carcinogen. That means it can cause cancer. The last but the most important thing while fumigation is being carried out is that a warning notice should be properly and visibly placed on the front door that indicates that the fumigation process is under progress, so that everybody is alerted that he or she should not enter that area because fumigation is being carried out. As I was earlier mentioning about some of the alternatives for formaldehyde, there are four other alternatives for the formaldehyde, like peracetic acid, hydrogen peroxide, chlorine dioxide, and ozone. Peracetic acid and hydrogen peroxide have some common advantages, like they are fast acting, sporocidal, that means they kill spores, they are non-toxic, they do not produce any residue, they disperse rapidly, and they are FDA approved, as well as they are cheaper alternatives. But the disadvantage of using peracetic acid is that it is also pungent in its odor, as well as it is temporarily corrosive. That means it corrodes where if it is used at high concentrations. The disadvantage of hydrogen peroxide is that it is not able to remove every trace of mycobacterium, and it is a very uh, strong source of contamination. The third one is chlorine dioxide. It has efficiency of killing almost all organisms, and it is non-carcinogenic too. But its disadvantages is that it can be broken down in the presence of UV light and it becomes corrosive. The fourth one is ozone. Ozone has the capacity of dispersing rapidly, but the disadvantages is it requires neutralization from time to time, and if it is used at high doses, then its efficiency also reduces. So this is about the alternatives of the formaldehyde, and let's have a look at how this particular fumigation process is carried out. So this is a video which I'll be playing for you all. In this you can see that they are mixing potassium permanganate with formalin. They have placed that beaker in the laminar airflow hood and they have closed the lid of that hood. So you can clearly see how the fumes are being generated out of it. And the personnel which are carrying out this fumigation, you can see that they are wearing their proper personal protective equipment like masks and spectacles, gloves, and lab coats. And you can see that they are sealing that hood properly with the help of the tape, and they are making sure that no traces of fumes come out of this hood. They are sealing it properly. Not a single point of leakage is left behind. Thank you so much for attending this session. And to complete this first session of fumigation, I would now request Ms. Shivangi Srivastava, who is a current student of MTech second year Biotechnology at School of Biotechnology, RGPV Bhopal. So over to Shivangi.
Hello everyone. I am here to complete the topic fumigation. As you know very well about the fumigation and pre-fumigation, now there is a time to know about post-fumigation. And what is the post-fumigation and why it is required? Now I am telling to you. A post-fumigation is the process where you can check the contaminants are still present or not after the fumigation process, which is very last and important step for knowing our environment is ready for further experiment or not. So for that, very first step is to be taken. There are some precautions which we should be taken, from which wear PPE kit before going to the fumigated area, because as earlier Riddhi ma'am described, there are some precautions which should be taken after the fumigation. When you are entering in the fumigated area, you should wear PPE kit, because some of the chemicals which are used in fumigation are carcinogenic and it can affect your health also. So always wear personal protective equipment, in which you have to wear compulsory. You have to wear lab coat, glasses, eyeglasses, gloves, mask compulsory. That will prevent you uh, from the further infection. After the fumigation, we have to check the plates in which chemical are used for fumigation in a way to avoid spillage. The image which is showing the black ash having the reaction completed after the fumigation, and this black ash is present. Now discard the plate carefully in the biosafety manner, because these chemicals which we were used for the fumigation are carcinogenic and may harm to your health very adversely. So you should consider that the plate should be, or the everything, each and everything that is used in fumigation have to be discarded properly in the biohazard bags which is available in the market. uh, for completion of post fumigation, there are some steps which have to be taken. The very first step is for checking the fumigation impact. We should perform an experiment which shows the variation in microbial load. As in pre-fumigation, we perform the experiment where we check the colonies came in the plates. So now we have to check the variation in both of the experiments, in pre-fumigation and after fumigation, so we can relate then with the fumigation is done properly or not. For that, we have to prepare agar plates and then incubated the plates in the fumigated area and in the fumigated lab. After 24 to 36 hours, colony will appear on the plates. So for that colony checking, you have to count colony by CFU, the colony forming unit. So now, if post fumigation is not done properly, then what will be the consequences you have to suffer from? There are some consequences. It may occur. It may hinders the results of your expected experiments that you want. That you want the expected results from the experiment, and contaminants that are still present after the fumigation, which should be harmful for you also and your health also. Eh, this will increase hard work while performing the experiments, because in biotech each and everything should be clear in your mind, and each and everything uh, should be noted out. So it will increases the hard work of your experiments. And there are so many wastage of hazardous chemicals, equipments, media and money takes place. So for as I mentioned you that there will be performing an experiment. So how to check the contaminants are still present or not? We have to do experiment from where we should prepare the media, as Archana ma'am told you about that. How should we prepare? How should be plating? How should be placed? And pour the media into a septic condition, that is in laminar flow, because laminar flow is providing you the contaminated free surface. And please wear the protective equipments like gloves. If you are touching directly the agar plate, that will also hinder your result. As you are saying that post fumigation is also experiment, so it will hinder your results too, because your hands also contain microorganism. If you are pouring the agar and your hands microorganism will grow on that, then how should you know that fumigation, your post fumigation is done or not properly? So place the plates in the lab where fumigated were performed. Some of the microorganisms will grow in 24 hours, and some of them take 36 hours for showing the colonies. So we have to preserve them for 24 to 36 hours. How to pour the plate? Here I put this photo in which the person who is pouring the plate wears the gloves. That means he don't want any hindrances in their experiment. For colony count, as I mentioned, colony count is necessary for that, because if you are going in a lab, okay, then you are inhaling and exhaling. So some microorganisms also transfer from you, because you also contain many of the microorganisms. So there will be need to know which microorganisms or which microbes will hinder, which contaminants will hinder your experiment or which are not. So for that we have to know which microorganisms are still present in our lab or which are not. So for that CFU is required. CFU refers to the colony forming unit, which is the number of an individual colony of any microorganism that grow on a plate of media. This value in turn represents the number of bacteria capable of replicating as they have formed colonies on the plate. So for checking that, we have to use this formula for the calculation. How can we check which colony of microorganism is present or which is not? So for fumigation, there are some data interpretation. Like for bacterial interpretation, there must be showing less than 10 colonies on a plate, then it will be applicable and there is no need of further fumigation, and now your lab is ready for the further processes and further experiments. Okay, but in fungal interpretation, you have to need that only 2 colony or less than 2 colony is growth on your agar plate. If it will increases from 2, that means your lab is also needed some fumigation again. This requirement is just for laboratory work. But if you are going to if you going to fumigate the OT, that is operation theater, you should mention in your mind that more than 19 colonies is not acceptable. You have to do fumigation if colonies of bacteria is coming more than 19. If 20 come, then you should go for the fumigation again. So here is the results after 24 and 36 hours. We have to check our agar plates. Here we checked, there are only 1 to 2, 3, not more than 10 colonies are found. So your lab is ready for the aseptic condition, and now you are going to experiment. You are going to do your experiment as well. If colonies growth of the microbial colony appears like this much of colonies are appearing, that will show the negative results, means still contaminants are present, and that will also hinders your results as well as your health. So for eliminating the contaminants, we have to repeat the process of fumigation. So there are some precautions which should be taken in the process of post fumigation. First of all, PPE kit should be wear before entering the fumigated area, and when you are working on agar plates, you are preparing the agar plates, you have to wear that PPE kit also for just removing the concern of microbial contaminants. Glassware should be sterilized for the post fumigation, because there will be some contamination present on glassware. If you did not sterilize, then it will hinder so your result also. Pouring the media into petri plate, that should be done in laminar hood, as I explained earlier. UV light should be switched off before using the laminar hood. Generally we are doing experiments, so we have just हम भूल जाते हैं कि हमें लैमिनार यूवी भी बंद करना है लाइट, तो it can also cause some harmful effects on your health. Maintain the other chemicals which should not place in the fumigated area, otherwise chances of explosion may occur. So you have to take these precautions while you are performing the post fumigation process. Now, thank you so much. Now I am inviting to Dr. Professor Archana Tiwari ma'am, which is the director of School of Biotechnology, RGPV Bhopal, to sum up the session first. Thank you.
Thank you so much, Ms. Shivangi. Wonderful presentation. In this session, we have discussed the three important things or the three important steps. That is, first one is pre-fumigation. Second one is fumigation, and third one is post-fumigation. Whenever we are talking about the pre-fumigation, what are the three basic characteristics or three basic things which we have to keep in our mind always? That are sealability, notification and warning notices, inform the staff concerning staff these lab is not ready to work under aseptic environment. Just check these labs, then only start the work under aseptic environment if the labs are ready. If not, just paste the notice that these labs are under observation, and first we will fumigate it, then only do all the process of pre-fumigation, fumigation and post-fumigation, then only the work begins, otherwise not. To inform the concerning officer, concerning staff, and finally ventilation is totally off of that area where we are expecting contaminants. First is sealability, second is notification and warning notices, inform the staff, and finally ventilation should be off of that area which is under observation for fumigation. So these are the pre-fumigation characteristics as we discussed earlier. Now, Riddhi Tiwari has discussed about the fumigation. The characteristic features of the fumigation are prevent excess, trained staff, specified procedure, operate remotely, and monitor frequently. Whenever there is requirement, not only because of the long COVID-19 pandemic, but also whenever we are working, there is a need of frequent fumigation to just protect our labs from the contaminants, because we are doing n number of experiments, n number of microbes we are carrying himself or herself, right? That's why there is a need of frequent fumigation of our labs to develop the aseptic environment or aseptic conditions. And finally, whatever we did in the two steps, pre-fumigation and fumigation, now we have to check whether these steps are up to the mark or not in the post-fumigation. That is, how much contaminants have already been removed. What is the level before we are entering in the lab? That is the level of fumigants. And we have to record all these, because when we are doing this, what is the need next time when we have to do this, we must know. That's why fumigation is the most important and foremost important step when we are going to start our lab work. In this manner, we have just completed our first session, that is the session of fumigation. If anybody is having any queries, kindly raise your hands so that we can entertain his or her queries. Anybody is having any query related with the fumigation, pre-fumigation, fumigation, and post-fumigation? If anybody is having any query, shall we proceed to the second session if nobody is having any query? Shall we proceed to the second session? Yes, please. Please, please.
Madam, we can use ethanol also for the aseptic conditions.
You are mainly using the ethanol for the sterilization of hands, sterilization of gloves, sterilization of our glasswares, not the complete lab, because ethanol is very, very costly also. We can do, but that is very costly as compared to formaldehyde. Any other question? Any other query? No query?
Ma'am, what is the time duration of fumigation?
Depends upon the number of load of your contaminant. Right? If you are using too much of microbes and you are not mainly taking the safety precautions, then what you have to do? You have to check in once in a month or once in 2 months. Depends upon the nature of the work which you are going to do, which you are doing. The next session is the session of cleaning of the glasswares, sterilization. For this, I am inviting Ms. Aditi Parekh, MTech 4th semester student of School of Biotechnology, Rajiv Gandhi Pradyogiki Vishwavidyalaya, Bhopal, to just throw some light on the process of cleanliness of the glasswares. Ms. Aditi Parekh.
So, thank you, ma'am, for such a wonderful introduction. And I'll be discussing about the how to clean the glasswares properly. And this is so nice to be connected to too many people at a single time since the social distancing hit us. So, let's start it. Good laboratory procedures require appropriate cleaning and sterilizing process to ensure excellent lab results. Despite being an important step in all the experiment processes, the task of cleaning of glassware is considered too mundane to be paid attention to. Dirty glasswares are the one of the most important reason behind the experimental errors. Often glasswares may appear clean, but they may be chemically dirty. Traces of ions, soluble substances, and solvent causes trouble that can be prevented with paying some extra attention towards the cleaning and rinsing process. Soap, detergents, and abrasive powders can be used to remove stains from a particular glassware. There are minimal requirements to properly conduct the cleaning process. All we require are a large clean sink area with undisturbed scenario to allow glassware to drain and to set glassware. Use a non-phosphate based detergent, some cleaning agents for some extra stubborn stains such as chromic acid, aqua regia, alcoholic KOH, etc. Some glassware can be dirty, they may contain grease. So grease can be removed by boiling in a weak sodium bicarbonate solution, soaking in potassium dichromate, rinsing with water followed by acetone and drying. One important thing to be kept in mind that never ever wash your glassware using a hand based soap solution or the hand cleaners because they often contain lotion additives that can form an oily film over the glassware that would further hinder our experimental processes. Next we require are the brushes depending on the size and the shape of the glassware, distilled water, sink drain, and some protective equipment such as rubber gloves because the cleaning agents may be corrosive and can cause damage to your skin, so rubber gloves are mandatory. Safety clothing and safety glasses. Usually the glassware that are supplied to us, new glassware that are supplied to us are sterilized, but due to some handling or storage or transportation error, mishaps may occur. So therefore, before using them, we need to clean them properly. For this, soak in 2% HCl solution for overnight to neutralize any alkali present. Wash the glassware in running tap water. Boil them in a synthetic detergent for 30 minutes and rinse it well with tap water and then a small distilled water rinse. For the used glassware, for all the glassware such as amazing cylinders, uh, what we say, beakers and flask, the process is same. Boil them in a detergent for 30 minutes. Clean them thoroughly with a brush provided. Rinse in tap water and then dry them in hot air oven with temperature not exceeding 80 degrees centigrade. Now let us discuss about some specific glassware that are regularly used in our laboratories. First one is the test tubes. For cleaning test tubes may if we have cultured in the test tube or if we have some infected material in the test tube, they need to be autoclaved before cleaning. For this purpose, boil them in for this purpose first we have to autoclave them in a autoclaving machine, then we have to boil them in a detergent solution for 30 minutes, clean with a brush thoroughly inside and out, and rinse them in running water, finally with a distilled water rinse, and then place them upside down on a rag and dry them in oven. So let me show you some pictures regarding this. These are some pictures. In the first case, we have just put some acetone in a cotton swab. Then we have put the cotton swab in a test tube. Use a tweezer to swirl the test tubes properly and then wash it with tap water followed by distilled water. In the next case, what we can do, we can pour some water to it, then add some detergent, shake it well and drain it, and then again wash it with running tap water. You can also use a tissue paper to gently wipe the inside of the test tube. For the pipettes, soak the pipettes in a cleaning solution for overnight. Wash them in running tap water. Then we have to rinse it with distilled water. And the suction pump of the pipette should be cleaned thoroughly because this may be the most contaminated part of the pipette. For this, dry on suction pump using spirit or acetone, and we can also sterilize if we need to. We can also sterilize the puppets by just plugging a cotton wool in their mouth and wrapping it in paper and then sterilizing it in hot air oven for 1 hour. Slides and glassware. Slides are one of the most used glassware in laboratories, starting from the schools to the colleges and in many institutes also. Usually slides are used for preparing the specimen of plants and animal cells, so they may be washed properly with glacial acetic acid for 10 minutes, and then or we can also wash them with dichromate solution for overnight. Then the process is same: rinse with distilled water, wipe them dry, and after wiping them dry we can either put them in jar full of alcohol to keep them contamination free, or we can dry store them and wash them in alcohol before we have to use them. So these pictures are just showing what I have just said. We are cleaning it and then we are drying them on tissue paper or wiping them with a tissue paper. Burettes. So one important thing to be kept in mind before we are cleaning a volumetric glassware such as burettes or volumetric flask, we should never use inappropriate brushes because these brushes can these crash the glassware and can cause an error in the calibration of that particular glassware. So for the cleaning of burette, we have to disassemble the we have to disassemble its organization. For that, we have to remove the stopcock or the rubber tip and wash the burette, as we have already discussed, with the soapy detergent, then with water, and then with distilled water to remove all the dirt or the contaminating materials. And we should also wash the stopcock or rubber tips separately, and before assembling them we can lubricate the joint. The next important glassware is most frequently used glassware is cuvette. So before moving forward, I would like to mention a point that the most infected part of any glassware are the rim or the edges or the bottom of the glassware. So special attention to be paid to those areas when we are just rinsing or scrubbing them. So mm, it becomes completely decontaminated. For the cuvettes, if the cuvette is not in the use right now, we can simply put some distilled water in it, wash it, and then we can blot dry them on a tissue paper. But if we are performing an experiment and we have to wash it in between the phases, so what we can do, we can just put the aqueous solution in it we can just put the blank solution in the cuvettes, followed by then decanting, and then followed by several washing with water, and we can dry them and blotting them on tissue paper. For the culture tubes and flask, we generally use culture tubes and flask or petri dishes for the culturing purpose, as the name suggests. So they may be sterilized before cleaning. And for this sterilization process, the best method is to sterilize culture tubes is by autoclaving them for 30 minutes at the 121 degree centigrade for 15 psi pressure. And we all know this is the standard protocol. After the media after the media turns to liquid on heating, which on autoclaving, we should just pour it out of the tubes. After the tubes are emptied, brush it with the detergent, water, and rinse thoroughly with tap water. Then with distilled water, and just dry them in a hot air oven at 60 degrees centigrade and place them separately. For the Petri plates, the process is completely same as of that culture flask or culture tubes. We just have to, after washing the Petri plates, we just need to sterilize them. So for the sterilization process, we have to wrap them in a heavy paper, and then we can put the Petri plates in the autoclave and then sterilize them at 121 degrees centigrade for 20 minutes at 15 psi pressure. Another important glassware is micropipettes. These are most widely used, and for cleaning the exterior part of the pipette, we only need to take some acetone in a small beaker, put the tip of the micropipette in that solution, and let it sit, let it sit on the pipette for 10 to 12 minutes before wiping it off. For the interior cleaning of the micropipette, we just have to disassemble the whole structure, and this can be done by reading the instruction manual provided with the micropipettes. And after disassembling it, we can use a long stem cotton swab with a dab of cleaning solution to clean out air and remove any clogs. We should also clean and rinse piston with distilled water and replace them if they are chipped, pitted, or broken. Lightly grease, lightly grease or lubricate the O-ring with the lubricant provided, and reassemble all the parts and check it for it if it operates smoothly or not. Some other important points I would like to mention are, if the rinsing and cleaning with the soap solution and water is not sufficient, we can also use some other uh, cleaning agents such as alcoholic KOH or aqua regia or We can also wash them with the acid solution such as 10% HCl or 10% H2SO4 depending on the glassware we are using or the contaminant it contains. So, thank you, and I would like to invite Ms. Yusra Siddiqui, who is a doctoral fellow at School of Biotechnology, to take over the session, and she'll be discussing about the sterilization and autoclaving process.
Good afternoon, everybody. Now we are discussing about the sterilization, mainly focusing on autoclaving, packing, and drying. In this session, we are going to discuss sterilization, methods of sterilization, autoclave, hot air oven, and precautions. As we know, sterilization is the process of eliminating transmissible microorganisms from media and the surface of the lab equipment. Sterilization is an absolute term which refers to any process that eliminates, remove, kills, or deactivated all forms of the life and other biological agents, such as fungi, bacteria, biological agents like spores, viruses from the given object. Absolute in the sense the given object is 100% free from the microbes, not accepting the range between 0 to 100. Sterilization methods are into two parts, that is physical methods and chemical methods. Physical methods are dividing into further dividing into two parts, that is heat and radiation. Heat is considered to be the most reliable method of sterilization of objects. Heat as a moist dry and dry most common sterilization methods used in different areas, which is indicating for the most materials. Heat sterilization further dividing into two categories, that is dry heat and moist heat. The dry heat sterilization, killing or removal of all microorganisms, including bacterial spores. This techniques required longer time exposure and a higher temperature than the moist heat. Various available methods of dry heat are hot air oven, flaming, red heat, etc. Dry heat ovens are used to sterilize items that may be damaged by moist heat or that are not impenetrable to the moist. For example, powders, petroleum products, sharp instruments, etc. See, dry heat sterilization causes the denaturation of the proteins and oxidative damage of the microorganisms. In the red heat type of sterilization that commonly uses for the sterilize the straight wires, bacterial loops, and spatula. In the flaming, also this sterilization techniques also uses for the bacterial loops, wires, and spatulas sterilization. And incineration techniques that is used for the solid soil dressing, pathological bedding, etc. And the last one is the hot air one. Moist heat sterilization are more efficient in contrast to the dry heat sterilization. It causes coagulation and denaturation of the proteins of microorganisms. The moist heat sterilizations characterized into three categories, which is based on the temperature. So, sterilization which occur at the temperature below 100 degrees centigrade, that is posturation, vaccine bath, serum bath, etc. Sterilization at which occur at the temperature of 100 degrees centigrade, boiling, steaming, And the last one is sterilization, which occurs at the temperature above 100 degrees centigrade, that is autoclave. Another type of physical method, that is radiation. There are two types of radiation, non-ionizing radiation and ionizing radiation. In the non-ionizing radiations, wavelength longer than visible light only considered. The UV radiation has a wavelength of 200. As we know, the UV radiation wavelength is the 200 to 280 nm. It has a germicidal effect on the microorganisms. So this is commonly used for the surface disinfection in hospitals, operation theaters, labs, etc. Ionizing radiation also having two types. That is particulate or electromagnetic. In the particulate sterilization, we use the electron beam, which is used for the sterilization of the instruments such as syringes, gloves, dressing packs, foods, pharmaceuticals, etc. Electromagnetic type of ionizing radiation uses the gamma rays or X-rays for this purpose to sterilize the disposable petri dishes, plastic syringes, antibiotics, vitamins, hormones, and fabrics. There is some limitation in heat sterilization, like they are not suitable for those objects which are heat sensitive and may get destroyed in the uh, pressure or heat. So, to overcome such limitation or problems, we use the chemical method of sterilization. The chemical methods of sterilization including the use of the following chemicals: alcohols, aldehyde, phenolics, gases, etc. So, alcohols basically dehydrate the cells of the microorganism and disrupt their membrane and causes the coagulation of their proteins. For example, ethyl alcohol, isopropyl alcohol, methyl alcohol, etc. The another one is aldehyde. Aldehyde acts through the alkalization of the amino acid, amino groups, carboxylic group, hydroxylic group, and probably damage the nucleic acid. It kills all microorganisms, including spores, for example, formaldehyde. Third one is phenol. As we know, phenol act by disturbing the membranes of microorganisms. They penetrate, they precipitate their proteins and inactivate their enzymes. So their examples are 5% phenol are mainly used for this purpose. Dettol, 5% Lysol, and the last one is the gases. So different types of gases are used in the sterilization technique. So that is ethylene oxide, formaldehyde gas, beta propiolactone gas, that is BPL. Now we move into the autoclave. So autoclave is a type of moist heat sterilization technique. This sterilization technique done with the steam under the pressure. Minimum time required is 30 minutes at 121 degrees Celsius at 115 psi pressure. That is the most reliable method of sterilization, that is rapid and effective for the sterilizing glasswares, plastic tubes, media, pipette tips, solutions, etc. So we can say the autoclave is a container similar to a pressure cooker. It is filled with the object to be sterilized and then sealed. Next, high temperature steam is forced under high pressure, thereby displacing air. Moist heat destroy microorganisms by irreversible coagulation and deterioration of their enzymes and protein structures. The time and the temperature of accomplish this depending on the pressure and the type of the microorganism to be inactivated. After the necessary time be passed, the steam is released and the sterilized objects are removed. The entire cycle can take from 15 to 16 minutes. The more commonly used methods when extended heat is not a concern is used to an autoclave or pressure cooker. When sterilization in this way, samples are placed into a steam chamber or a shell or a rice floor, and chamber is closed and heated so the steam forces air out of the vents. Pressure is then applied so the interior temperature reaches to 121 degrees centigrade or 250 Fahrenheit. This temperature is maintained for between 15 to 30 minutes. This elevated temperature and the pressure are sufficient to sterilize samples of any commonly encountered microorganisms or spores. So as we know, hot air oven is commonly used for the dry heat sterilization, and dry heat sterilization is a method of controlling microorganisms. It employs higher temperature in the range of 160 to 180 degrees centigrade and require exposure time up to 2 hours depending upon the temperature employed. Okay, the benefits of dry heat includes good penetrability and non-corrosive nature, which make it applicable for the sterilization glasswares, metal, surgical instrument. It is also used for the sterilization non-aqueous thermostable liquids and thermostable powders. Dry heat destroy bacteria and endotoxin which are difficult to eliminate by other means, and this properly make it applicable for sterilizing. Dry heat kills by oxidation, protein denaturation, and toxic effects of elevated level of electrolytes. How can we use the hot air oven? Articles to be sterilized are first wrapped or enclosed in a container of cardboard, paper, etc. Then the material are arranged to ensure uninterruption airflow. Oven may be preheated for the materials with poor heat conductivity. The temperature is allowed to fall to 40 degrees centigrade to remove all the sterilization materials. Sterilization protocol. First, transportation of the contaminated equipment. So, isolating the used and contaminated equipment from the already sterilized equipment in order to not to contaminate them, and then go for then the cleaning of the equipment by using manually or chemical method should be performed in a design area immediately after procedure. Quick cleaning removes residues much easier and minimize instrument staining, corrosion, etc. The another one is inspection. So in addition, check each lab equipment for proper function and lubricant those that are metal to metal actions. Packaging. Sterilization wrap may be used for the packaging instrument sets for an effective barrier. To maintain a sterilizing unit, be sure to double wrap per wrap. After that, the sterilization. As we discussed previously, sterilization can be done via the means of steam, chemical, vapor, rapid heat transfer on the lab equipment. Then go for the experiment conduction. Experiments are now equipments are now sterilized and contaminant free. So we are ready for experiment. Precautions. So do not seal containers under pressure while autoclaving as they may explode. Always use personal protective equipment that is PPE when using an instrument. Wear a lab coat, heat resistant gloves and safety gloves. If you suspect leakage or any other problem with the instrument, do not operate the instrument or machine. Immediately contact to the responsible person. Make sure the equipment are dry before keeping them in hot air oven. Let the oven cool down before 2 hours before opening the door of the oven. Always follow the given instructions to the appropriate instrument. Thank you so much. I would like to invite Aditi to continue the session.
So, to sum up the whole cleaning and sterilization process, this is a pictorial representation that gives an idea about the whole process of cleaning followed by sterilization in the same manner as they are done. I won't go in the details as we have already discussed everything, but to sum up the session, following are some important key statements we should keep in our mind. It is very important to clean and sterilize our glassware before executing an experiment because it may give error, erroneous results if we use dirty glassware. Likewise, presence of impurities in glass labware can distort the meniscus and can prevent one from getting the correct result out of the experiment. Keeping the laboratory glassware physically neat and clean, free of grease, dirt, chemical, and metallic residues and bacteria is therefore of the utmost important. One has rightly said cleaning and sterilization of glassware is just like cleaning your weapons for the next battle. Thank you. And now I would request our director ma'am to take over the session and share the concluding remarks.
Thank you so much, Aditi, for a wonderful presentation. You and Yusra both. Now, this is the time just to wind up today's both the sessions. The first session of fumigation and the second session of cleaning of the glasswares and sterilization. Whenever we are talking about the fumigation, as we discussed in the fumigation session, how it is important, why it is important, what is its significance, and how it is very, very basic or before beginning of the experiments, we have to check whether our labs are having the aseptic conditions or not. In the second session, we have discussed about the cleaning of the glasswares and their sterilization. Whenever we are talking about the cleaning of the glasswares, equally important as it is as important as the fumigation. Why we are saying this? Because whenever we are cleaning our lab, but we are not cleaning our glasswares, it makes no sense. So whenever we are starting our experiments, we have to very cautious about the lab, the belongings of the labs, and the experiment which we are going to conduct. In this manner, we are going to end today's first and second sessions. Tomorrow we are going to start the third and fourth session. Third session mainly contains the preparation of the solution, volume by volume and weight by volume of the stock solution and the working concentration. That is the working solution. Till then, we are meeting tomorrow at the same platform, at the same time. Till then, always take the vitamin P. P stands for positivity. Now we are ending this workshop's first day sessions, first and second, and leaving the meeting. Thank you very much once again to all the participants for joining this workshop and making this workshop or making us very happy for providing us such type of platform. Once again, thank you very much from the bottom of our hearts. On behalf of the School of Biotechnology, we are just thank you one and all. Thank you one and all. Thank you. We are leaving the meeting now. Tomorrow we are again meeting at the same time, at the same place, on the same at the same time, on the same platform. Till then, be safe or stay safe and stay healthy at your homes. Thank you very much.